Hello, everyone. Welcome to our lecture on weeks 3 and 4 for Microbiology Lab. We're going to talk a little bit about a couple of different experiments that you are learning about this week, and I just wanted to provide a quick lecture and some overview for you just to help you prepare for your midterm practical. We already covered immunological testing in class, but I will cover it again in here just so that you have a recording of it and you're a little clearer. So in immunological testing, the first experiment that we look at in immunology is our titer. And a titer is used to determine if a patient already has pre-existing immunity to a specific disease. The type of titer that we are using in our laboratory is called an Agglutination double diffusion test. It uses a small little plate. That tiny plate has some agarose in it, and the agarose has no nutrients or anything. We're not growing any bacteria, but we are going to drill little holes. So we have this kind of round plate like this, and then in the center, we're going to create a well. So there'll be a hole in the center of the plate. It's very small. It only holds a few microliters of solution. Then at the top of the plate, we usually have some more holes that surround it. We go all the way around like this. And in the center well, what you can see here, in the center well where it's labeled A, is where we're going to place some soluble antigen. Now, this is the Agglutination double diffusion test. It uses the property of diffusion. And in diffusion, when we add a soluble antigen, that's what our immune system would react to. We create antibodies against an antigen. When that antigen is soluble, we have it in solution and we put it in the center well, and it's going to diffuse throughout the agarose. And it diffuses, you can see here, it's going to diffuse in all directions at the same rate. That's what they're showing in this close-up here on the right. Then we are going to take some patient serum. Patient serum is obtained by taking a blood sample, spinning out the red blood cells, and then collecting the serum or plasma that is left behind. The plasma contains the soluble antibodies that we are currently testing for. Most antibodies, with the exception of one class, are soluble. They are dissolved or soluble in the patient serum. So we're then going to add patient serum to each of the 5 wells. But when we add this patient serum, we do it through a series of dilutions. So in the top well, we're going to have undiluted serum. So this is straight patient serum directly from the blood sample. We've not diluted anything. As we move around the wells, as we move around the plate, we have dilutions. So here we have a 1 to 2. This would be a 1 to 4, 1 to 8. 1 to 16, and 1 to 32. So what you should notice here is that every single time we dilute the patient serum, we do so in half. So we just keep cutting it in half and then in half again, in half again, in half again. And what this really means in numbers is if I have 100,000 antibodies, if my patient has 100,000 antibodies in this 10 microliters of their serum, then technically my next dilution should contain 50,000. Right? So this should have 50,000 antibodies in it. And this one should have 25,000 because this is 1/4 the number of antibodies as the undiluted. So the idea is that we just continue to dilute the patient's serum. As diffusion occurs, we have diffusion of our antigen occurring, but we also have diffusion of the antibodies. They're going to be diffusing in all directions at the same rate as well. And when we have enough antibodies bind to their antigen, we get what's called an antibody-antigen complex. This complex becomes very large, and it will precipitate out of solution, just like any chemistry precipitation result. So our antibodies and antigens come together. That's what they're showing you in a close-up here. And all of these antibodies bind to lots of antigen, and it creates this big giant kind of matrix-like complex, and it becomes too large to remain soluble. It diffuses out of, it precipitates out of solution and creates this white line, this white chalky looking line, and that's referred to as a precipitin arc. So it's a precipitate. Here is a picture of our of a plate from our actual laboratory. This is the result of the experiment that is described in your lab manual. And this is from our actual laboratory. And you can see this patient has a reaction at undiluted, so they've certainly been exposed to this particular antigen. But their titer is a 1 to 2, right? So this patient has a 1 to 2 titer, or a titer of 2. If we take a look over here on the top set of boxes from left to right, we have the order in which this reaction is going to occur. So we take our patient sample and we put it in a serial dilution series. We then expose it through this diffusion process in the agar, and then we look for that precipitation reaction for each patient dilution. In the set of boxes below, we have what the different readings mean. So in this case, if my titer is a 1 to 16 or higher, so my patient would have to have a reaction all the way up to this titer dilution right here. If it's 1 to 16 or greater, meaning 1 to 32 or 64, etc., then my patient is considered to have sufficient immunity. If my patient has enough antibodies in 1/16 the dilution of an undiluted titer, then my patient definitely has enough antibodies to be immune to that particular disease. They have enough antibodies that if they were going to come into contact with that antigen, they would be able to destroy it before it could ever take hold and cause disease. If their titer is 1 to 8 or less, meaning it's only up to here, if it's 1 to 8 or less, they have some immunity, but it may be considered insufficient. So we would certainly suggest that the patient receive a booster vaccine. We want to say, hey, listen, you have some immunity. There's obviously some memory cells there, but we need to boost it. We need to really kind of make sure that your immunity is very strong. So we would give a booster vaccine. If there's a titer reading of 0, meaning not even the undilute, this undiluted here, there's no reaction at all. We don't see any precipitin arc anywhere on the plate. This indicates that the patient has never been exposed. If the patient has never been exposed to that disease, they're not going to produce any antibodies against it. That person is totally susceptible to that particular disease. They have absolutely no resistance. So for this particular test in this Agglutination, right, this is called that Agglutination double diffusion, we certainly want to make sure that we know what our different titer cutoffs are. 0 is unexposed. 1 to 8 or lower or less is insufficient, needs a booster. 1 to 16 and higher is considered sufficient immunity. There are lots of different types of titer tests out there. You may learn about the hemagglutination titer, other types of titers, but this is kind of one of the originals out there, this double diffusion method. These pictures here at the top are just kind of more diagrams of the same thing, kind of showing you where these different types of reactions are. Here we can take a look at this one up here. This patient has a 1 to 16 reaction. That's what this drawing indicates. So this patient would have sufficient immunity, whereas our patient in the photograph below does not. So this here is the Agglutination double diffusion or immunodiffusion titer reaction. In the review questions, what type of reaction is occurring in the plate that allows us to observe this reaction? This would be B, our immunoprecipitation reaction. And in question number 2, does this patient require a booster based on their titer reaction? Well, we can see that the highest titer number that we have in actual precipitin arc, that's what that is right there, is 1 to 8. So this patient, we would recommend them to receive a booster vaccine. The next immunology test that I want to talk to you about is a passive agglutination test. Now, agglutination is used when we are looking for antigens that are insoluble, that are not dissolved in solution. An agglutination test, a prime example of an agglutination test is blood typing. Blood cells are not soluble in patient serum, red blood cells. So in blood typing, this is a form of agglutination test. In this particular instance, we are doing this with immunological compounds or proteins and antibodies. So this would be an immunoagglutination test. In this particular test, we are going to be running a test for what's called CRP. CRP stands for complement reactive protein. When you cover your immunology chapters in your lecture course, you will learn this as C3 convertase. This is the complement protein that C3. C3 is elevated. We produce C3 in elevated levels during inflammatory conditions. It's also produced in elevated levels during bacterial infections. And CRP is unique in that one of the effector functions of the C3 convertase activation of CRP activation is actually destruction of gram-negative bacteria cells. So it makes sense that during bacteria infections we would have an increase in CRP production. But the problem we have in testing for CRP is that CRP is produced by our own bodies. This means that we are not going to produce antibodies against it. We don't naturally produce or normally produce antibodies against our own proteins. This means that we must use what's known as an indirect agglutination test. We must introduce human CRP to some other cell culture or another organism in order to obtain antibodies from a different source. We're not going to get antibodies from our own serum because we don't produce antibodies against our own proteins. So an indirect agglutination test is when we get antibodies from another source. A direct agglutination test is when we use our own antibodies. So CRP is considered an indirect agglutination test. In the CRP test, we are looking for an actual agglutination or clumping type reaction. Now, since we have to obtain antibodies from another source, the companies that create these test kits, these are usually purchased as a kit. This is an actual diagnostic CRP kit that we use in our laboratory. We use the latex agglutination test. So in this particular test, these small, tiny little microscopic latex beads are coated with antibodies, and the antibodies have a general Y shape, and the stem region here is constant, and the antigen binding sites are on the ends of the Ys. So these are adhered to these microscopic latex beads. I don't know how these companies do this, so I can't answer that question, but I can tell you that the antibodies are bound to these beads so that their antigen binding sites are sticking out. We then expose the patient's serum to these this solution containing these microscopic beads, and if our patient is producing high CRP levels, we'll get this agglutination type reaction where they all start clumping together, similar to an antibody-antigen complex. Below is a result from a CRP test. So here in the middle is our patient sample. On the right is a negative control, and on the left is a positive control. And in this particular test, we always put our patient sample in the middle so we can compare it to our two controls. You can see the patient sample is very milky. We have a small collection in the center, but for the most part, it's very milky and kind of opaque, very similar to the negative reaction. On the left is a positive control, and the positive control has a very rigid kind of outline, and it's kind of grainy. We can see the agglutination is really certainly starting to occur in this particular sample here. So in this case, we would consider our patient to be negative for elevated CRP levels. If we take a look at our review here, let's start with question number 1. If the construction of the CRP test or how a CRP test works. So if the construction of it makes it an indirect agglutination test, how would we classify blood typing? Well, the CRP test, we have to get antibodies from another source from outside of the human body, so that's indirect. In blood typing, we actually use human antibodies. If you have type A blood at birth, you naturally produce antibodies against type B. So we would have direct agglutination if we were looking at blood typing. Question number 2, what result in the image below indicates a positive reaction? Definitely this is going to be A. We see all of this clumping and graininess. It's really grainy inside of the liquid here, whereas B is very milky and opaque. So B is negative, A is positive. So I'm going to go ahead and end this video here. Again, this is just a review on the immunological tests. Hopefully this is something that you find helpful. And a copy of the PowerPoint will also be uploaded into the D2L course shell. See you in the next one.